Hello and welcome to this new Genetic Engineering and Biotechnology News webinar entitled Fibro-Inflammatory Disease Sees Relief in IL-11 Monoclonal Antibody Development. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Biologic drugs have dominated the biopharmaceutical market in recent years, and there continues to be no shortage of therapeutic targets for drug developers designing monoclonal antibodies. Recently, since IL-11 has been identified as an essential cytokine for inflammation and fibrosis, investigators have been hard at work developing monoclonal antibodies toward this protein to be used for the treatment of a variety of conditions such as non-alcoholic steatohepatitis or NASH. Findings have shown that several antibody clones are highly potent inhibitors of IL-11 signaling and block the transformation of fibroblasts or hepatic stellate cells into myofibroblasts. Additionally, several preclinical models of fibrotic disease showed that antibody treatment prevented and even reversed fibrosis while preserving organ function. Let's meet our speakers for this webinar, who will tell us the benefits of IL-11 antibody production and the clinical relevance of this antibody for use in vitro and in vivo studies of liver disease. Stuart Cook is a professor at Imperial College and Duke National University of Singapore, as well as co-founder of Enleofen. Sebastian Schaefer is an assistant professor also at Duke National University of Singapore, as well as a co-founder of Enleofen. Stuart and Sebastian will be co-presenting data about the newly developed IL-11 monoclonal antibodies and their effect on treating fibro-inflammatory diseases such as NASH. Before we begin Stuart and Sebastian's presentation, I want to encourage the audience to submit questions for our Q&A session immediately following the talk. We'll try to get to as many questions as we can, so simply type your question into the Ask a Question box on the right-hand side of the screen and hit submit. All right, with all that said, let's begin our webinar. Gentlemen, the floor is yours.
Hello everyone, welcome to this webinar about first-in-class IL-11 neutralizing antibodies, which we've put together between Enleofen and Aldevron. So my name is Stuart Cook. I'm a professor at Imperial College and Duke NUS in Singapore and co-founder of Enleofen.
And I'm Sebastian Schaefer. I'm a geneticist originally from Germany, and I'm now also working at Duke NUS and for Enleofen Bio. We founded Enleofen in April 2017 as a spin-out from Duke NUS and SingHealth, and licensed all the IP we generated around IL-11 and the antibodies into the company. Enleofen now has taken over the antibody development and ensures that we have a good path forward to the clinic for our molecules.
So on this slide, we want to give you just a brief overview of the biology behind the company and our products and where we're heading to in the clinic. And so if you look at this slide now, on the left-hand side, what we're showing you is some results from our paper which was published in Nature in 2017. And what we're depicting here is the human genome from the first chromosome to the X chromosome on the X-axis, and on the Y-axis you're seeing RNA expression. And this is fold change compared to baseline. And so what we did for this experiment is we harvested a piece of tissue, heart tissue, from patients who are undergoing cardiac surgery. We then grew out the cardiac fibroblasts from those biopsies, and we did this in approximately 80 individuals. And then we studied these fibroblasts either when they were quiescent and unstimulated, or after they've been stimulated with TGF-beta, which you will be aware is a very strongly pro-fibrotic cytokine. And we asked the question, could we find something downstream of TGF-beta that was important for activating the fibroblasts? And could we find something that was actually distinct from TGF-beta itself? Because as you may be aware, targeting TGF-beta is toxic and has limitations as TGF-beta is redundant. So we were looking for something that was non-redundant and was hopefully safer to target. So if you can see now, the colors of the circle show you the correlation of the gene expression with ACTA2 expression, which is the transition of a fibroblast into a myofibroblast, and the size of the circle shows you the p-value. And without any clever statistics, you can just see that out of all the genes in the genome, there on chromosome 19 is a huge increase in the cytokine called interleukin 11, which has on average about an 8-fold increase in its gene expression in the activated fibroblasts and correlated with fibrosis. So this is very surprising to us because we'd never heard of this gene IL-11 and certainly knew nothing about its role in fibrosis, which we'll talk about a bit more in a minute. If you look at the right-hand side, you can now see some data from the Phantom Consortium. This is CAGE data, gene expression data. And we asked the question, what is the difference between IL-11 receptor expression compared to that of IL-6 receptor? People are more familiar with IL-6. IL-6 is a very important inflammatory cytokine, and its receptor, as you can see here on the right-hand side, is highly expressed in inflammatory cells, as you might expect. However, if you look at the IL-11 receptor, it's most highly expressed in a completely different set of cells, for instance, the fibroblasts and adipocytes, but also epithelial cells, and very rarely do you actually see both receptors co-expressed in a cell. And so it's almost a mutual exclusive situation here, where you have IL-11 receptor expressed on stromal cells and fibroblasts and epithelial cells, or you have IL-6 receptor expressed when you're looking at inflammatory cells. Here on this slide, what we're showing you, depicting, are the publications for either IL-6 or IL-11 since their discovery. And you can see the publications for IL-6 in blue have been very many. It's a very well-studied gene. In fact, it's one of the top 10 genes studied of all time. However, in contrast, IL-11 has hardly been studied at all, and you can see it depicted in red. The reasons for this are quite a few. We'd like to highlight one, and that's that IL-11, when it was first discovered, it was discovered in fibrocyte-type cells from the stroma of the bone marrow, and was found to support megakaryocytes, probably indirectly. And for this reason, people made the incorrect assumption that IL-11 was somehow linked with megakaryocyte function and platelets. And at that time, a drug was developed called Numega, which is recombinant human IL-11. And if you inject that into mice or monkeys or humans, you do actually get an increase in platelet count. This actually is an off-target effect, and it's due to the fact that individuals receiving IL-11, whether you're a mouse, a monkey, or a human, get global organ fibrosis, including fibrosis of the bone marrow stroma. And if you have a fibrotic bone marrow stroma, then you have a reactive platelet count. The platelet counts go up due to this off-target effect. So unfortunately, people thought that IL-11 was fully understood and it was important for platelets, but it absolutely isn't. The knockout mouse for IL-11 receptor has normal platelets. Knockout humans for IL-11 receptor have normal platelets. And if you give our antibodies in long-term therapy studies, the platelets are normal. And this is one of the reasons why we believe IL-11 has not been studied very much, is people thought it was a done deal when it very much wasn't.
And we would also like to highlight that there are very severe side effects for the treatment of Numega or recombinant IL-11, and these side effects are also indicative of global organ fibrosis that is occurring in these patients.
So what we're showing you now on this next slide is a fish. And the reason for showing you this is we went back in evolution to try and properly annotate the function of IL-11, and we've done so here using the fish as a model. So if you go back to the fish, you find that IL-11 is most highly expressed in stromal and systemic immune organs when the fish is exposed to a pathogen. And you can see on the right-hand side here on the gel that if you inject PBS into a fish and you profile for IL-11, you don't see it. But if you inject a bacteria, you see very robust induction of IL-11 gene expression, and here in the liver and the head kidney at 24 and 48 hours. And that is in response to the bacterial associated molecular pattern, the PAMP, as they're known. And this is actually inducing IL-11 expression in the immune system to fight the infection at the stromal epithelial interface. And so there's a lot more data on this, but we won't show it today. But IL-11 is very important for the immune function at the epithelial stromal interface, and its expression is not in immune cells at that interface, It's in the epithelial cells and in the stromal cells. So now here on slide 7, just giving you a very high level overview of what we now have come to understand about IL-11. As we showed you, we first discovered this in the cardiac fibroblasts, and we published this in 2017, where we showed it was extremely important for fibrosis. However, what we've come to understand now is also critical for stromal inflammation indirectly. So this is IL-11 activating stromal cells to then produce pro-inflammatory cytokines like CCL2 and CCL5, which then activate and encourage immune cells into the tissue. And then on the bottom right-hand side here in the grey, you can see we also discovered that IL-11 is very important for the parenchymal function. So that is, if you have an epithelial cell or a polarized cell, so for instance a tubular epithelial cell in the kidney or a hepatocyte in the liver, if those cells get exposed to IL-11, they become dysfunctional and they're no longer able to perform the correct function of that organ. So in the liver, the cells can no longer package fat properly, and in the kidney, they can no longer work on excreting urea and creatinine. And so this now gives you a reason why inhibiting IL-11 is so critically important for disease, because we have very large effects. We're hitting 3 different aspects of the tissue level disease. Yes.
So this is some data that we generated before we actually started the antibody development. And this is in part also from our paper in 2017. So when we discovered IL-11, we first generated the or actually received the IL-11 receptor knockout animals. These animals, as Stuart already mentioned before, are actually completely fine. They don't get infections. They have a normal lifespan. However, we then realized, running different preclinical fibrosis models, that these animals are, as we would expect, protected from fibrosis. So on the top left is a model of kidney fibrosis. Actually, it's the folate model. We have animals on the left-hand side are wild-type animals, on the right-hand side are knockout animals, and we measure the collagen content here with a quantitative assay, the HbA assay, and you can appreciate that receptor knockout animals have reduced collagen content in the kidneys after folate treatment. In the middle is a model for cardiac fibrosis. This is the TAG model. Here we constrict the aorta and induce a very a thickening of the heart muscle together with fibrosis. Again, we can measure the collagen content in the entire heart, and we see a reduction in the collagen content in the receptor knockout animals. This is confirmed with a second cardiac model, which is an angiotensin II induced cardiac fibrosis model. Again, we see a reduction in the collagen content. We actually also started to explore a bit the other models and tissues, skin fibrosis, but this in general was very difficult because there's several disadvantages in using a genetic knockout model. So the first one is that if you have this knockout strain, these knockout animals are actually not completely normal. At baseline, we see there are differences in gene expression, for example, and this is likely due to the fact that these animals, even though they are fine, have somewhat adjusted to a loss of IL-11 signaling throughout their lifespan, so they are basically not normal. The second problem is that if you have a genetic model, you're really fixed to a certain genetic background. And as I'm sure you're aware, is that different mouse strains will react differently in different preclinical models. So in some preclinical models, you cannot use this strain. The third problem is that you're not really very flexible in your experimental design. For example, it's not possible to do a late intervention study where you first let the disease develop and then later on treat the disease. Of course, another big question remains is that is this target that we're investigating here actually therapeutically accessible? So is it possible to translate this into the human patient setting by, for example, using a therapeutic agent such as an antibody? So this was all the reasons why we then started to look into therapeutic antibodies or antibodies also for use in our in our lab. And then we contacted Aldevron, and we really enjoyed the interaction with them, so we started a whole program, actually 2 programs. And Aldevron performed their genetic immunization of mice, which was really proved to be quite powerful, because with their approach, they actually managed to generate several antibodies for us, some that would bind IL-11, some that bind the receptor, and what we were very surprised to find was that they actually managed to break the immune tolerance of these animals, and these antibodies actually also bind to the mouse antigens. We did these 2 campaigns because we wanted to make sure that the pathway that we're investigating is really the critical pathway. So we got cytokine and receptor antibodies to make sure that we have specificity in the program, and we also were interested to see if these 2 approaches phenocopy each other. In this program, several clones were discovered and screened also initially for some specificity of binding to our antigen, and then we received several clones and tested their efficacy in the lab. So then there were actually quite some challenges on our side, because as Stuart already alluded to before, IL-11 was largely mischaracterized. And this was not just evident from all these clinical clinical programs that were happening and also in vivo observations, but actually also in vitro assays that were associated with IL-11 function to us seemed very wrong. So there is one example on the left side. This is a cellular assay that was used to look at IL-11 function in vitro. And what is really weird is that in this case, for example, you need about 250 nanograms per mil of IL-11 to induce a cellular response. So this is actually completely, that is a completely different ballpark to what we are seeing. We can use IL-11 in the very low nanogram per ml or even lower range and see a very strong cellular effect. So we think that in these examples there is actually unphysiologically high concentrations of the cytokine, and this is not the real biological function of the cytokine. If we screen for antibodies inhibiting this interaction, we think it's not reflecting the true biology. It's also very difficult to get a reporter assay, because the signaling complex of IL-11 has many subunits and is very difficult to translate that into an assay. So what we did was we looked at our results from our studies, and we developed a tailored cellular assay, so actually several tailored cellular assays to better understand the IL-11 effect and how we can effectively neutralize the IL-11 effect. And what for us was very important to identify the most potent clones was that we used the relevant cellular system with the cells that are important for disease. We actually used different types of stimuli, so not just IL-11, but for example, we also used an upstream stimulus of IL-11 to make sure that we can block endogenous as well as exogenous IL-11. And we also focused on readouts of the cells that are very important for disease. And then on the using these cellular systems, we did perform several screens. We also performed screens with different species to identify cross-reactive clones, and scored all of the different clones that we received to identify our top hits. And so this we brought together antibodies from 3 different campaigns into Enleofen. 2 of those were done together with Aldevron, and this was the campaign ENX203, which is mouse monoclonal antibodies binding IL-11, ENX campaign, a campaign ENX209, which is mouse monoclonal antibodies binding to receptor. And we also integrated from a phage display campaign a human antibody binding to IL-11, which we term the campaign ENX108A. 108A, yes. So this is the end result of many years of work. And this is some example data of the IC50s that we generate using our cellular assays. In this instance, you're seeing two different stimuli. One is IL-11, one is a cell stimulated with an upstream cytokine of IL-11. So we're blocking both endogenous and exogenous IL-11. And here we're titering down the antibody concentration using a clone that we received from Aldevron. And as you can see, this clone is highly, highly potent. So we can go down into the low nanogram per mil concentration and still block significant effects that are induced by IL-11, either endogenously or exogenously. And this is really an industry-leading IC50 efficacy in the picomolar range.
So now moving on from the antibodies a little bit to use in vivo. So we're not going to be showing you in vitro data, of which we have a lot, but we're going to go straight in vivo. So showing you first here that we like to use the approach of both genetic loss of function and then combined with pharmacological loss of function to really confirm specificity of the effect that we see. So on the left-hand side, and again, this is the limitation as pointed out by Sebastian of using a knockout, that you can only do in a constitutive knockout a prevention experiment. And so that's what we're showing on the left-hand side. And so we've taken wild-type mice, which is the white and the grey circles, and put them onto a Western diet with fructose supplementation. So this is the McDonald's and Coke diet. And you can see if you put a wild-type mouse onto this diet, there is a very robust increase in collagen in the liver. And then below that, you can see an increase in the serum ALT. So this is a marker of liver damage, of hepatocyte cell death. Then if you look at the effect in the knockout, so these are the black circles or the blue circles, you can see that baseline the knockout is the same as the wild type, but when you put the knockout onto the Western diet, you actually don't have an increase in collagen, and your serum ALT levels are markedly lower than that of the wild type mouse. And so that shows you very beautifully that in the knockout and prevention experiment, that there's a very profound effect of removing the IL-11 signaling on accumulation of collagen in the liver or liver damage. Obviously, what we wanted to do then was the therapeutic experiment, and we're showing you this now on the right-hand side. And here we put wild-type mice onto a Western diet for 16 weeks. They become very obese, they become hyperlipidemic and hyperglycemic, insulin resistant. And after 16 weeks on this diet, we then start them on antibody therapy. And we've done this with both of the antibodies that we did with the Aldevron campaigns. And you can see here we then treat the mice for 6 weeks with either an IgG, which is the blue circle, or the anti-IL-11 antibody. In this case, I believe it was the 203 campaign. And as you can see, there's actually a decrease in the collagen compared to 16 weeks in the anti-IL-11 treated animals, whereas collagen actually continues to go up in the IgG treated animals. And after the 6 weeks of therapy, there's an over 50% difference in the collagen content, which is an accepted readout by the FDA for studies of NASH. What we're showing you on this slide is something that was really quite surprising to us, but was hinted on the previous slide. So we knew that IL-11 was important for fibrosis, as we discussed. That's how we found it. But on the previous slide, we showed that it was also having an effect on the ALT. So that tells you that somehow inhibiting IL-11 signaling is actually protecting the hepatocytes. So in this experiment we're showing you now at the top, on the left-hand side, you can see we put these mice onto this very toxic NASH diet called the high-fat methionine choline deficient diet. And after 1 week, the ALT, which is the liver damage marker, has gone up from about 40 to about 700. So they get very severe acute onset NASH. We then start either IgG therapy or anti-IL-11 receptor therapy and continue that for another further 5 weeks. In the middle panel at the top, you can see that as expected, the fibrosis does not develop Here, this is more of a prevention experiment with the antibody. So fibrosis does not develop when we inhibit IL-11 signaling. However, the IgG does a very robust and week-on-week increase in the fibrosis of the liver. But what's really quite remarkable is if you look on the bottom left now, is the effect of therapy on liver damage. And so as I mentioned, after 1 week on this very toxic NASH diet, the livers are pouring out during necroinflammation a huge amount of ALT at 700 units per litre, and that is then reversed to normal within 3 weeks of anti-IL-11 receptor therapy, whereas the IgG continues to go up over that time, up to about 1,000 over the same period. So this shows a very profound effect of anti-IL-11 receptor therapy on protecting and regenerating the liver. On the middle panel there, we're also now showing you the effect of the therapy on the function of the liver, so the function of the parenchyma, and that's the ability of the liver to package and export triglycerides and utilize triglycerides as a fuel source. And so in the IgG treated animals, there is a very large increase in the triglyceride content in the liver, whereas in either those animals receiving anti-IL-11 or anti-IL-11 receptor therapy, there is a very large decrease in the triglycerides in the liver. And you can see this at the histological level, so looking now at the micrographs on the right-hand side in either ENX203 treated livers or ENX209 treated livers, they look normal. But if you look at the IgG livers, they look like very, very fatty and dysfunctional livers, which is exactly what they are. So you can just see at the macroscopic level a very profound effect of anti-IL-11 therapy on the function of the liver in addition to its anti-fibrotic effects.
So now at the very end, I want to highlight another a very important aspect of using antibodies in university as well as in the company setting, and that is intellectual property. So when we discovered IL-11, there was basically no knowledge about IL-11 in fibrosis, and the pre-existing literature actually suggested an opposite effect, a protective effect of IL-11 in disease. So for us, it was actually quite easy to argue that all of our findings were novel. Now, what was the absolute key was that we then had antibodies available to show that blocking IL-11 signaling was therapeutically beneficial. And this has led to a number of patents. The first patents have now actually published and have also been already granted. And I just want to highlight some independent claims that we have received in the US as well as in Europe. And these claims actually give us protect the field for us from competitors, because there was no pre-existing literature. We have been able, using these antibodies, to claim any antibodies binding to either IL-11 or the receptor could be a therapeutic agent to treat numerous fibro-inflammatory diseases, which is very powerful and is not possible also to do with a genetic model, because you in the end have to prove you can really use an antibody to interfere with the signaling. So to finally come to the conclusions, I think what we have shown to you and what we want to tell you in this webinar is that antibodies really provide a superior platform for both in vitro and in vivo preclinical studies, and are very useful actually in the academic setting as well. That you can generate highly relevant, clinically relevant data that goes far beyond what is possible using genetic models. They provide an unprecedented flexibility in the preclinical studies. We can do early intervention, late intervention, regeneration. All of this would not be possible without antibodies. This data can then be used as a basis for intellectual property, and in the end, if the program is successful and you generate a lot of data, then actually you can directly use these molecules to jumpstart the development of therapeutics by going into humanization programs.
Great. Thanks very much for listening. Look forward to the questions.
Stuart, Sebastian, that was a great presentation with some very compelling data. The clinical potential for these antibodies would seem very high, and I look forward to your future work. Before we begin the Q&A session, I want to let everyone know that this is one of their final chances to submit questions for our speakers, so hurry up and submit them now. So it looks like we've gotten a bunch of really great questions that have come in already. So let's get to the Q&A session. Just give us a moment to get everything coordinated on our end, and we'll begin the Q&A presently.
All right, we've got some really terrific questions already, with more still coming. And I'd like to mention, though, before we get started, that Stuart wasn't able to stay for the Q&A portion, but Andreas Weiser, also from Aldevron, has agreed to help out. So let's get going with our first question. And this first question, I believe, is for Sebastian, and that is there a need or advantage to targeting the delivery of IL-11 antibody in the treatment of NASH, or is systemic delivery sufficient to gain therapeutic effect?
Hello, thank you for the introduction, also for this question. So the short answer is no, there is no need for targeted delivery. This is actually true for NASH or for any other disease we have tried so far. And for this, of course, it's quite important that one has very potent antibodies. So in the beginning of the program, we were really unsure whether or not this effect that we had discovered and verified using the genetic models would really be therapeutically accessible. And it is really required that a systemic administration of the antibody is sufficient to block the disease, because this is, of course, the clinically relevant route of administration. So we are hoping to develop these antibodies further to enable us to do an IV administration of the antibody into patients, or if in an ideal case possible, even a subcut administration of the antibody, because if you want to treat someone with NASH, chances are high that this person is not feeling this sick, and if you have to administer the antibody very often, if you have to administer it in the hospital, then actually this treatment is not really suitable, and it's not possible to translate our findings into the clinic. So with this antibody that we have shown to you today in the webinar, we know through a PK study and looking at the biodistribution of the antibody that about 10% of the antibody homes into the liver, and this is sufficient for us to block the fibrosis in NASH, the inflammation, as well as the hepatocyte damage in NASH, using a concentration that is actually that can be administered in a clinic as well, which is about around the 3 mg per kg administration, a systemic administration.
All right, thank you very much, Sebastian. Now, the next question I have is for Andreas, and I'll ask the question, but Andreas, since they didn't get a chance to meet you earlier during the event, you might also want to just give a quick introduction to yourself so everyone knows who you are. But first, the question they're asking is they're wondering why, one of our listeners is wondering why genetic immunization was selected or chosen.
Okay, thank you for the question. So my name is Andreas Weiser, and I joined the company Aldevron 2.5 years ago, and I'm mainly responsible for project management and client relations. Okay, so concerning the question, it's always a matter of question how you would like to immunize the animals depending on the later usage of the antibody. So in this case, you are looking for a functional antibody which probably is able to block a cytokine or a receptor. And one of the great advantages of the genetic immunization is that you can express the target in the native form in the animals. So the target is presented to the immune system in the animals in its native form. And if you, for example, choose peptides or proteins which are recombinantly produced, it could be that the conformation, or it's more or less clear that the conformation is not the native conformation. And by doing the genetic immunization, you can assure that the antigen is presented to the immune system in its native form. So this is the highest chance to generate antibodies which are really functional in vivo, for example, like in these cytokine blocking assays or something like this. So that's really one of the key strengths of the genetic immunization.
All right. Thank you, Andreas. The next question, I believe, is also for you, but if Sebastian would like to jump in on it, that's certainly fine as well. And that's one of the listeners is wondering if you have to pay royalties on the antibodies.
Okay, what I can say is that if we run an antibody project, this is completely CRO, client research organization things, and you don't have to pay any royalties. So you get the sequence of the antibody in the end, and we are just the developers when we don't have, so there's no licensing or something, because all the rights on the sequence then belongs to the customer or to the client.
Yes, something I just would like to add. I think talking about royalties, this is a bit more common if one orders or works, collaborates on generating human antibodies or generating generating antibodies with humanized mice. So with Aldevron, we were focusing on mouse monoclonal antibodies, and there is no immediate clinical relevance because there's still a lot of work needed to move those antibody assets into the clinic.
All right, thank you both. The next question, I'm not sure for whom this one is, but the next question is, do the commercially available antibodies replicate the findings of Enleofen assets?
So yes, I can say this is the case. So there is some neutralizing antibodies available. However, we find that those antibodies are far less potent than the lead antibodies that we have selected from the campaign. We have been, or Aldevron has been able to generate several antibodies, and they went through a very thorough screening. And the antibodies that you see the in vivo data with is made with those antibodies that are very potent neutralizers. The commercially available antibodies can be used to replicate the findings, but they are usually working only in vitro at rather high concentration.
All right, terrific, thank you. The next question is, are there specific subtypes of fibroblasts that are IL-11 producers versus those that are not?
Yes, so this is a very interesting question. So not every fibroblast is the same, and there actually are differences. So, but first of all, I would also like to point out that not only fibroblasts can make IL-11. We actually also found out that cells of the parenchyma, such as, for example, hepatocytes, can also produce IL-11, and they can also sense IL-11 because they carry the receptor just like fibroblasts. In terms of fibroblasts, all the fibroblasts have the IL-11 receptor. However, there is a certain subgroups of fibroblasts that specifically are characterized by an elevated IL-11 expression. And this has been found now in two studies that are not from us. One is a preprint on bioRxiv from the Broad Institute. This was published, I think, in October, so a few months ago. And in this case, the group studied patients suffering from colitis from UC. And as I'm sure many of you know that are familiar with the field, a big problem in UC is resistance to anti-TNF therapy. Actually, about 30 to 40% of the patients do not respond to anti-TNF therapy. What this group did was it performed single-cell sequencing of the colon of these patients, and it compared either responders of the therapy versus those individuals that did not respond to the therapy. And what they discovered was that there is a specific cell type in the colon of these anti-TNF resistant patients, and they term this cell type a inflammatory fibroblast. So what this fibroblast does is it's as the question asks, it's characterized by expressing very high levels of IL-11, and then in a secondary effect, driving the inflammation that cannot be stopped by blocking TNF-alpha. So in this case, it's a subtype of a fibroblast that is very important for inflammation. There's another study that just came out a few weeks ago, actually, from the group of Paul Noble. This was about IPF fibroblasts. So what they did was they performed a migration assay of fibroblasts from IPF patients. And in IPF, or actually also in other fibrotic diseases, one very characteristic feature of the disease is that the fibroblast can migrate and is very mobile. So what they did was they had a chamber where fibroblasts could migrate from one chamber into another one, and then they captured the fibroblasts which either migrated or didn't migrate, performed RNA sequencing, and again, I think in the top 5 or 10 upregulated genes in those fibroblasts that are able to migrate is again IL-11 as being highly upregulated in this fibroblast. So these two studies already show that fibroblasts that tend to be very important in disease, either through migration, collagen secretion, or through driving stromal inflammation, are actually characterized by expressing IL-11.
All right, thank you very much for that good, very detailed explanation. Our next question is, other than HPA, have you done any histological imaging and analysis of your diseased versus treated groups, and what are the findings like?
So yes, this is true. So we did some histological imaging, and these studies are actually just under review, and we will publish them very soon. Long story short, if we quantify fibrosis with staining and histological images, we can see the same findings as using the HPA. That being said, we really prefer the HPA assay because we believe it's the most accurate, since it really, first of all, is a biochemical molecular assay and is not based on quantification of images. And second of all, it's based on the entire tissue, because we grind the entire tissue and then perform the assay on that. And that is a very representative amount of collagen that's indicative for the whole tissue and not just for the section that you have a histological image from.
All right, thank you. Our next question is, what is serum IL-11 level in human and your NASH model?
So this is extremely low. Usually it's not detectable either in health or in disease. And the expression of IL-11 is really to detect that one has to go to the tissue level, look in the liver, and then look at the protein, for example, with the Western blot, to see if IL-11 is up. But this is usually not sufficient to elevate the IL-11 expression so it's visible in the serum of patients. In healthy individuals, IL-11 is basically not expressed, and there it's completely off in all of the tissues.
All right, thank you. Then the next question from one of our listeners is, do we have IL-11 receptors in megakaryocytes? And is there any research about oprelvacin-induced bone marrow fibrosis?
Yes. So this is a very good question because, as I'm sure some of the viewers will be aware, if you think about IL-11, most people actually think about hematopoiesis and about an effect on platelets. And this is because when IL-11 was initially discovered in the early 90s, it was discovered in a fibrocyte cell line that was supporting a hematopoietic cell line. And at that time, it was misinterpreted that IL-11 has an effect on the hematopoietic cell line and subsequently on the platelets. But this was reason enough for Pfizer to develop a drug called oprelvacin or Numega, which is basically recombinant IL-11 and which induces elevation in platelets in patients when it's administered. We believe this, as this person already points out, that this is actually because IL-11 causes a bone marrow fibrosis in these patients, and that results in an elevated platelet count. But it's not, we believe, it's not a direct effect of IL-11 on the megakaryocytes. So we are a bit looking into this because this is really a tragedy, I think, for the IL-11 field, because this has been the basis for many misleading studies that followed this, because it was somewhat considered that IL-11 is important for hematopoiesis and is actually a protective cytokine, whereas we believe the exact opposite is the case. And actually, if you look into the product sheet or in the side effects of this drug, you actually see that the side effects are very severe and they are indicative for causing a systemic fibrosis in these patients. These patients, for example, all have elevated BNP levels, which are indicative of the cardiorenal problem. Many of them actually, I think about 14% get get arrhythmias, which is probably due to fibrosis of the heart, and there are many other problems as well.
All right, thank you. It looks like we have time for one more question, which I believe is also for you, Andreas. And that is, what is the process for approaching the company with a research proposal?
Okay, yeah, thank you. Actually, I put this question because I wasn't sure absolutely who was or to whom the question addresses. So I just thought I explain how it works in our company if we do a research campaign or antibody campaign for any client. So it is basically very easy. You just can directly call us in Freiburg or write an email. Or there's also an online questionnaire available on our website, aldevron.com. And it's very easy just to clarify questions. And to start up a project, it's very easy. We can just start from the in silico sequence of the target. In case of the genetic immunization, which is our biggest business, so it's actually very easy to get in touch with us.
All right, thank you. We might as well squeeze in at least one more question, since I see we do have a few more minutes. And somebody would like to know, is asking, are you aware of the new publication in BioRxV showing IL-11 as a therapeutic agent for IPF? That's by P. Novo and Stuart Cook, Benjamin Eng et al. And they evidently use a tool antibody to block ILRA KO mice.
Yes, because that's actually us. So we have now done several studies, and this is a parallel study to our liver study. So this is actually also based on the antibodies that were generated by Aldevron.
All right, thank you. Then one more is, which antibody campaign gave you the final binder, Aldevron's campaign or the page display approach?
I can also answer this. So there is no answer for this. We have been pursuing both programs in parallel, and we see advantages in both programs. We see advantages from phage, we see advantages from starting with mouse monoclonal antibodies, and one can talk just about 1 hour only about this. What I think is very important is if one has the capabilities to have a broad program, actually use several tools and develop them in parallel, because there is always something that can happen throughout the program with any clone, and it's very important to have backup assets.
All right, thank you. And I think with that, we have come to the end of our webinar. I'd like to remind everyone that the webinar will be archived for 1 year on our website. So if you missed any parts of it, you can watch it again, or of course, feel free to forward the link to your friends and colleagues. I'd like to thank Stuart and Sebastian again for their informational and very informative presentations, and Andreas for assisting with the Q&A, and I'd like to thank the audience for their attention and thoughtful questions. And of course, a very special thanks to Aldevron for sponsoring this webinar. Hopefully we'll see all of you at another Gen webinar in the near future. Goodbye for now.